Hello everyone. Today I am going to take a seminar on the topic micrometry. Micrometry or morphometrics as the measuring of different dimensions, chromosomes, cells, tissues, and other minute and microscopic objects using a microscope. These measurements are based on the comparison of the objects with the standardized calibrated scales and are expressed in micrometers since microorganisms, cells, and such minute objects are visible only under a microscope. A suitable scale for measuring them should be within the scale itself. The device used for measuring microscopic objects is called a micrometer. A micrometer consists of two components, namely stage micrometer or objective micrometer and ocular micrometer or eyepiece micrometer. Stage micrometer is a rectangular glass slide with a 1 mm long graduated line scales engraved on it. It can be conveniently mounted on the stage microscope. The linear scale has 100 equal divisions demarked by parallel lines. Each division therefore measures 0.01 mm. Every fifth division on the scale from the beginning to the end is demarked by longer lines. Ocular micrometer is a transparent and circular glass disc or plastic disc within linear scale at its center. It is placed in the body of the ocular tube. The scale has 100 equal divisions demarked by parallel lines. Every 10th division from the beginning is demarked by a longer line. Ocular micrometer differs from the stage micrometer only in the value of the scale is not given. Calibration of ocular micrometer to stage micrometer. Micrometric calibrations involves finding out the absolute distance on the stage micrometer corresponding to one divisions of the ocular micrometer scale. From this, the value of one division in the ocular micrometer can be calculated. This value is called a micrometer value or calibration factor. Is done by only after superimposing the graduations of ocular and stage micrometers. Focusing of the stage micrometer will cause the superimposing of scale of the ocular micrometer over the scale of the stage micrometer. Now the value are compared and number of divisions on the ocular scale equivalent to each divisions of the stage micrometer is calculated. The ocular scale value is converted to stage micrometer value. During this operation, the ocular micrometer have been calibrated against the stage micrometer. It involves of the following steps: removing the eyepiece of the microscope from the ocular tube and note its magnifying power. Then unscrew the cap of the eyepiece and tightly insert a metal ring between the inside wall of the eyepiece tube. Then insert the ocular micrometer into the eyepiece tube until it comes to rest flat on the metal ring. Screw the cap of the eyepiece and reintroduce it to the ocular tube of the microscope, and then mount the stage micrometer on the stage of the microscope and focus in such a way that both the scales are clearly visible. Note the magnifying power of the objective lens, and then adjust the stage micrometer and the eyepiece in such a way that both the scale coincide and exactly overlap each other at the at the starting point. Observe the further ends of the two scales and note the point where they coincide and overlap each other, and then count the number of divisions between this point and the starting point of on each scale. To the calculation, the value of one ocular micrometer divisions is calculated with reference to the number of divisions on the stage micrometer by applying the formula V is equal to x by y into 10, where V is the value of one ocular micrometer divisions and x is the number of counted divisions on the stage micrometer, and y is the number of counted divisions on the ocular micrometer. Let the number of divisions on the stage micrometer be y and the number of divisions on the ocular micrometer be x. Since each stage micrometer division is 0.01 mm apart, the distance between the starting point and the point of coincide on the ocular and stage micrometer can be calculated by at that magnifications. Since one stage micrometer division is 0.01 mm and the distance covered by x divisions of ocular micrometer is equal to y divisions into 0.01 mm. One ocular meter division is equal to y divisions into 0.01 by x divisions. For example, suppose 10 ocular micro divisions overlaps 7 stage micrometer divisions at 40 magnification. One ocular division is equal to 7 into 0.01 whole divided by 10 is equal to 0.007 mm. Therefore, calibration is the process of determination of the magnitude of one divisions of the ocular micrometer at a particular magnification with the help of a stage micrometer. Here, the value of ocular micrometer division is calibrated as 0.07 mm. Since this value varies with the magnification of the objective lens, calibration of the ocular micrometer must be done from different objective and eyepiece lens. The actual length or diameter of a microscopic object can be obtained by multiplying the number of ocular micrometer divisions overlapped by the object with the calibrated value. For examples, in the above context, if the number of divisions overlapped by the specimen is 13, the width of the specimen is 13 into 0.07 is equal to 0.091 mm. Once the calibration is over, the stage micrometer is replaced by the microscopic slide containing the mounted materials which has to be measured. Then focus the lens so that the material can be seen superimposing on the ocular micrometer. The different dimensions of the materials are noted in terms of the number of divisions of the ocular micrometer scale. Then multiplying this number with the value already calculated to get the actual dimensions of the materials.